I would explain it as overall everything that happens in a laboratory so far that manipulates gene sequences, it is called genetic engineering. It's an overall term, okay? But that can be subdivided into different categories of engineering or technique. We have what we call the initial early genetic engineering, which most of us tend to associate now because it's been actively going on for longer, is what we call, and let's not get bogged down by the names, but it's called transgenetics, because we're taking a gene that we've isolated and spliced out of, usually initially it was some bacteria, gene sequence, and we forced that, often literally by shooting these genes into a completely different organism, okay, in this case a plant. So we would, what was used as an example early on was that we would take a gene from a toad and we would shoot it into a tomato. And something that the toad's genes were expressing would be helpful for that tomato, say, in either increasing the yield or preventing it from being eaten by a particular insect. So that's transgenes, completely different species into a completely different organism. Okay, so transgenes. The other kind that was developing in parallel is gene editing. So instead of literally taking a gene from one organism and putting it in another, it's more of making a chemical snip. They don't actually do it with scissors, they do it with chemicals, and they remove, isolate, copy, or prompt the expression of a trait within one plant, for example, or one microbe, one fungi, or one bacteria. So they're not bringing in foreign genes, they're just making alterations to a particular gene within that plant. Okay. Those are the two main categories. There are others, and it is a whole bunch more complicated than this, but this is, an effort to try and simplify it in a way that everyone can understand.